In this video we will discuss about the role of ALDH4A1 in the suppression of tumorigenesis. So far we have understood that ALDH4A1 form a trimeric complex with MPC1 and MPC2, thereby helps in the metabolisms in TCA cycle in the mitochondria. Here we will discuss if this trimeric complex get disturbed, uh, then it will affect whether it will affect the tumorigenesis or cancer proliferations or not. So for that they use different kind of approaches to disrupt this trimeric complex. First they use UK5099. This is the inhibitor for MPC, and here they have seen that when they use the UK5099, it will cause increase in the relative growth of the pancreatic cancer cells, and when they suppress this MPC1 or MPC2, there also they see a relative increase in the growth compared to the control cells. Thereafter they check the effect of ALDH4A1 knockdown on the glycolytic rate using ECAR, glycolytic reserve using ECAR, and there they have seen that when the ALDH4A1 is knocked down, then they see increase in the glycolytic reserve in the ECAR, that is in the cytosols. But when they reintroduce the ALDH4A1 wild type, and they can see a decrease in the glycolytic rate, and when they overexpress ALDH4A1, they can see a further decrease compared to the control case, indicating that high amount of ALDH4A1 can control this glycolytic rate. This glycolytic rate might be causing high glycolytic rate might be causing the high proliferations of the cells. So here in the panel they shows the immunofluorescence image, where what it is, they use the ALDH4A1 knockdown and they can see an increase in the glycolytic signals. But when they reintroduce ALDH4A1 wild type in the knockdown cells, they can again see a decrease in the glycolytic rate. Quantifications are shown in the right hand side, where we can see that the effect of this 2-NBDG, which is a kind of inhibitors, which is 2-NBDG's uptake at different conditions. So we can see that when the ALDH4A1 is knocked down, they see a high amount of 2-NBDG uptakes, but when the wild type is reintroduced, they see a low amount of 2-NBDG uptake, and which could be a markers for this, which is marker for this glycolytic rate in the cytosols. And thereafter they also check the proliferations, the panel lab, where they can see using different reagent. For glucose they do not see any changes. When the ALDH4A1 knockdown, they see high proliferations, but when they supplement with galactose or 2DG in the ALDH4A1 knockdown, they see suppressions of the proliferation of the cancer cells, indicating that this galactose and 2DG can play a role towards reversing this proliferations effect in the case where ALDH4A1 is knocked down. And further they check the effect if they knock down any of the one trimeric complex component of the complex, it is ALDH4A1 or MPC1 or MPC2. So they can see that when they overexpressed this ALDH4A1 wild type, they see a decrease in the proliferations, but when they knock down either MPC1 or MPC2, they see increase in the proliferations. And when they reintroduce in the for ALDH4A1, they can see some suppressions compared to the case where MPC1, MPC2 is knocked down. And the last yellow line indicates the conditions where MPC1, MPC2 is knocked down, but ALDH4A1 is overexpressed. And these things they have validated using this panel K, where they shows the Western blot or of overexpression of ALDH4A1 and the knockdown of MPC1 and MPC2 in the vector case, and also in the case of where this ALDH4A1 is overexpressed. Next, what they did is they checked the in vivo studies. They did in vivo studies and checked the tumor size, tumor grade, tumor volume. So in the case where ALDH4A1 is overexpressed, they see a minimum amount of tumor growth compared to the case where MPC1 and MPC2 is knocked down and ALDH4A1 is not overexpressed. They see a high tumor growth compared to the case where ALDH4A1 is overexpressed. And these things they have quantified on the panel J, where or the time dependent results are shown on the panel J, where we can see that the case where the all the three components of the trimeric complex is knocked down, that is MPC1, MPC2 is knocked down, not all the three, but MPC1 and MPC2 is knocked down, and ALDH4A1 is at the base levels, it is not overexpressed. At that case they see a high tumor volume, but when it is overexpressed they do not see too much changes, or the changes are not statistically significant compared to the case where they do not make any changes, or in the control they see some tumor volumes. In the minimum tumor volumes they see where ALDH4A1 is overexpressed and MPC1 and MPC2 are also present. Next, they check the relative growth rate, and the relative growth rate also is minimum when the ALDH4A1 is knocked down. When the ALDH4A1 is overexpressed, not knocked down, when the ALDH4A1 is overexpressed, and it is actually minimum in the case where MPC1 and MPC2 are present, and also there is no changes done in the ALDH4A1 compared to the case where ALDH here the results for the ALDH4A1 overexpressions are not there, but for the case where ALDH4A1 is knocked down are shown here, and when the MPC1 MPC2 is overexpressed. So when all the threes are present they see a minimum growth compared to the case where any one of them are absent or knocked down. So finally what they have able to find out is that this glycosylated pathway is converted into the pyruvate, and thereafter the pyruvate will be utilized by MPC1 MPC2 ALDH4A1 in the mitochondria in the TCA cycles. However, if any of this trimeric complex component of this trimeric complex is not working, or it is knocked down, then it will cause a high glycosylasis in the cytosols, and that will help in the tumorigenesis of the cancer cells. This is what they have finally come up from the results that are presented in this figure, and also the results that we have discussed in our previous videos. So I hope you understood how they come up to a point that ALDH4A1 helps in the suppression of tumorigenesis. If you need to understand more about these experiments in detail, you can go to the reference that has been provided in the description. That's all for this video. Thank you for your time.